Okay, so now I'm gonna talk about the difference between rods and cones in our eyes. Rods have this name because they are an elongated cell body that gives it a rod shape. A rod is a photoreceptor, which is a specialized type of nerve which has the role of taking in light and converting it into a neuron impulse. Inside each rod, there are thousands of structures known as optic discs, which are large membrane-bound structures. In the membrane of each optic disc, there are many proteins which takes in light and causes the rod to release an action potential that will soon reach the brain. On the other hand, a cone gets its name because it is cone-shaped. It is also a photoreceptor that has the same internal structure as a rod, with optic discs and proteins embedded in them. While the proteins in the rods are called rhodopsin, the proteins in the cones are called photopsin. When a ray of light enters the eye and hits a rod, and happens to hit a rhodopsin, it will turn on the phototransception cascade that will cause the rod to release an action potential into the brain. In cones, this process is exactly the same, but it involves photopsin. In addition, in an average retina, there are 120 million rods and 6 million cones. So, if I draw a very simple structure of an eye, with this being the optic nerve and this being the retina, rods are found in the periphery of the eyeball, so in this area and in this area. In the retina, there is a region that goes towards the inside, which is called the fovea, and cones are found right next to it, about here. While cones produce colored vision, rods do not because they are 1,000 times more sensitive to light than cones. Therefore, the light detection in rods results in a black and white vision, while the light detection in cones results in colored vision. Rods take longer to release an action potential compared to cones, and cones are able to quickly adapt to changes in light, while rods take much more time.